My name is Mariya Sweetwyne. I'm an assistant professor here in the Department of Lab Medicine and Pathology, for those of you who don't know me. And I'm really excited to introduce Dr. Kurt Zimmerman, who is a friend, a colleague, and also a scientific relative. So Kurt and I went through the same lab at UAB for our PhD. And he's originally from Wisconsin, and he did his undergraduate work at the University of University of Wisconsin. Oh, sorry, wrong UW in Eau Claire. And then he went to UAB, where he joined the lab of Joanne Murphy Ulrich. And I did not like Kurt for a while. I'll tell you why. So I'd never met him. I graduated before he started, but I kept in touch with my graduate mentor. And the way she would gush about this guy in emails, like, oh, this new graduate student, and he is just so focused, and he's so great, and he's so wonderful. And you know, I was not that, so I was pretty resentful. But she was not wrong. Kurt won the Joint Health Sciences Outstanding Graduate Student Award at UAB when he graduated, which is like there's no higher honor that you can get as a graduate student. And then he went on to do his postdoctoral work with Brad Yoder. And for those of you who don't know Brad, he's also at UAB, but Brad was really sort of instrumental in determining that polycystic kidney disease was a ciliopathy, was a disease of actually dysfunctional cilia in the kidney. And so Kurt went on to do his postdoctoral work with Brad, was super productive, which was no surprise to anybody, and then started his own lab at Oklahoma Health Sciences Center in 2019. And another funny connection between us is that, so his graduate thesis actually sort of built on my graduate thesis in calreticulin regulation and collagen, which like nobody cares about now, but and then somehow we both have ended up in the kidney. And so I've had the pleasure of getting to know him. And it turns out he's like a really nice guy, even if he is like super great at stuff and stuff. So he's a good person to talk to and a good person to know, and a really excellent and collaborative colleague. In addition to running his own lab, where he'll talk about the work that they're doing there, he's also the director of the Immunology and Informatics Core at Oklahoma Health Sciences Center, which he just informed me is a name that he made up himself. And so without further ado, I hope that you all enjoy this talk. I know it's going to be great. And Kurt.
All right. Can anybody hear? Mic on? Sir? Okay. Well, thank you, Mariya, for that kind of embarrassing introduction. But was very much appreciated. Thank you for the kind words. So also thank you, Mariya and Scott, for the invitation to speak to you all today. Had a great time visiting with different faculty members throughout campus today, and it's graduate students, and it's been a really outstanding day of conversation. So today I am going to be sharing with you all a couple of stories that we've generated whereby we use single cell transcriptomics to study immune cell function in the context of our favorite disease, which is polycystic kidney disease. So as Mariya mentioned, I actually came from Brad Yoder's lab, and Brad is a very passionate cilia biologist. So for those of you who are not familiar, primary cilia are these small microtubule-based structures that protrude from the cell surface of almost every cell type in the body. They're composed of this 9+0 microtubule doublet structure, and it's thought that they're involved in sensing changes in the microenvironment. And when they detect those changes, they transmit the signal down through the primary cilia into the nucleus to alter gene transcription. Now, if you take a look at the tissue that we're predominantly interested in our lab, the kidney, using a cilia reporter mouse in which every single primary cilia is labeled green, you can see here that the tubular epithelial cells in the kidney each contain a single primary cilia, and that primary cilia protrudes into the tubular lumen, where, as I mentioned before, it's thought that it senses the tubular flow. So for example, if you take this same cilia reporter mouse and you actually stick it on a microscope and you do some live intravital imaging, what you'll see is that these primary cilia are all oscillating in the direction of fluid flow. So you can see that they're oscillating back and forth, and that matches with the direction that the fluid is flowing through the tubular lumen. Now, if you take and you do some sort of injury or disrupt the normal kidney homeostasis, what happens is those primary cilia now flop back and forth really rapidly. And we think what happens is that change in the fluid flow within the tubular lumen is sensed by the primary cilia. They bend back and forth, and then it transmits signals down through the primary cilia to the nucleus to say, hey, something's messed up, something's going on in the kidney. We need to alter our transcription and proteins that we're producing to make it better. Now, like I said, the function of the primary cilia is beginning to become appreciated, but one thing that's really well understood is that if you mutate genes that are required for normal cilia structure or function, and I've listed some of those shown here on the left, it results in a spectrum of phenotypes that are referred to as ciliopathies. Now, ciliopathies can include things such as polydactyly or excess digit formation. It can include situs inversus or flipping of the organs. And the one that our lab is predominantly interested in studying is the cystic kidney disease. So cystic kidney disease is caused by mutations in those genes that are required for normal cilia structure or function. The most common of which, some of you may or may not have heard of, are the mutations in the PKD1 or PKD2 gene, which cause the autosomal dominant form of polycystic kidney disease. However, I always like to highlight, because this is something we'll get dinged on in grant applications sometimes, is that cystic kidney disease can be caused by a variety of different cilia-related mutations, which has kind of been, as Mariya mentioned, like the hallmark of Brad's work in his time as a faculty member. And I've just listed some of those other mutations that can cause cystic kidney disease shown here as well. Now, collectively, these disorders affect about 1 in 500 people, and as the name would imply, is characterized by these fluid-filled cysts that are present in the kidney and the liver. And this is a picture of a kidney that was isolated from an AD PKD patient. And you can see here the large fluid-filled cysts are these little circular-like things. And then you also develop a lot of interstitial fibrosis, which is the white part of these kidneys here. Now, these patients also will develop extrarenal manifestations. About a third of the time, they'll get a liver phenotype. And you can see here, compared to the normal liver here, these patients will get these massively expanded livers, which can cause portal hypertension, ascites, and some of those other issues with the liver. Typically, the liver problems won't necessarily kill you, whereas the kidney problems will, due to the development of end-stage renal disease or end-stage kidney disease. Treatments for polycystic kidney disease are fairly palliative in nature, and that means that the way that we treat polycystic kidney disease is we put patients on dialysis, or we transplant the kidney, we give them a new kidney. I will say that the first FDA-approved treatment for polycystic kidney disease was approved a couple of years ago, and it's been shown that it significantly enhances the lifespan of these patients and improves their quality of life. So I think in the PKD field, we're starting to make some headway into developing therapeutics for this disease, but there's clearly a lot of unmet need at this current time. Now, if you look at the cellular mechanism of how a cyst actually forms, despite the fact that this is called a dominant disease, which typically means if you inherit one mutated copy, you get the disease, that's not necessarily how the autosomal dominant form of this disease is inherited. So what happens is these patients will inherit one normal copy of the PKD1 or PKD2 gene and one dysfunctional copy. And it's thought that the development of cysts requires a second hit mutation. I will say we have some ideas what the second hit is, but we may not 100% understand what it is at the current time. But what happens is when you get that second hit in the normal copy of the PKD1 or 2 gene, you get a haploinsufficiency, so you get a reduced amounts of PC1 or PC2 dosage, and that causes these epithelial cells with that second hit to dedifferentiate, to proliferate, to secrete fluid into the tubular lumen to produce chemoattractants. And you can see here that these cells lacking the PKD1 or PKD2 gene or having reduced PKD1 or 2 dosage begin to proliferate, they begin to expand, the fluid gets pumped into the tubular lumen here. They produce factors that recruit other immune cells and other effector cells such as macrophages, T cells, fibroblasts, and then you can imagine over the course of time that this just continues and continues and continues until you get something like I showed in that picture before. Now, if you look at the molecular mechanism of cyst formation, this is a really oversimplified version of what's going on in these kidneys, but basically what happens is that you get reduced PC1 or PC2 dosage, that's the protein names for PKD1 or PKD2. It causes activation of adenylyl cyclase 6, resulting in cyclic AMP production, pKa phosphorylation, and pKa phosphorylation is kind of a hallmark of PKD. It causes the ion channels such as the aquaporins to be inserted into the apical side of the tubular lumen, as well as it drives cellular proliferation, resulting in that cyst expansion, what I kind of mentioned to you earlier. Now, it can also cause production of NF-kappa or activation of NF-kappa beta, which results in production of these macrophage chemoattractant cytokines that bring in those immune cells to modulate the rate of cyst growth. Now, one of the really interesting things about patients with PKD is that even if you inherit a mutation in the PKD1 or PKD2 gene, the rate of progression in these patients is really variable. So what I mean by that is if you look at these three different graphs that I'm showing you up here, and you look at cyst volume or kidney volume is shown in the y-axis, and age is shown in the x-axis for all these graphs, you can see here that a certain cohort of patients have really rapid cyst progression over the course of a few years. And this is, once again, despite the fact that these patients have a mutation, they all have a mutation in the PKD1 or PKD2 gene. Now, in contrast to this group of patients, there's a second group of patients you can see here on the bottom that have relatively stable cyst growth and kidney volume growth over the course of several decades. So some are progressing over the course of several years really rapidly, and some are really stable for several decades. And then these can be broken down clinically into class 1E or class 1E or A based on the Mayo classification system. Now, a significant amount of work from our lab and others have shown that there's several different factors that can influence the rate of cyst growth, not only in patients, but also in mouse models. A list of some of these factors that are proposed to influence the rate of cyst growth is found here on the bottom. This can include the identity of the gene that's mutated. So typically patients that have a PKD1 mutation will progress more rapidly than a PKD2. The type of genetic mutation, you can imagine this is a dosage disease. So if you have a missense or a nonsense mutation, it can affect the dosage of the polycystic 1 or 2 protein. Sex, genetic modifiers, so mutations in other cilia-related genes can influence how severe the disease is in this patient. And then the one that our lab is predominantly interested in studying, which is environmental modifiers or kidney injury. So we really think that kidney injury is a modifier of the rate of cyst growth in human patients. The majority of data supporting the role of kidney injury as an influencer of the rate of cyst growth comes from mice models. And you can see I've put up here two different studies, one done by Jing Zhao's lab and one done by Dr. Yoder's lab at UAB, showing that if you give mice with mutations in PKD1 gene, shown here on the left, a kidney ischemia reperfusion injury, it greatly accelerates the rate of cyst growth. Likewise, data from my former lab, Dr. Yoder's lab, show that if you give a mouse with a mutation in the IFTD8 gene, kidney injury, it greatly accelerated the rate of cyst disease. So as of today, the majority of data supporting the role of injury in accelerating disease has come from mice. And to my knowledge, there hasn't been a clinical study that's been published to date. So I often get that question, so I just like to get out in front of it here. So one of the big overarching questions of my lab is trying to understand what is the involvement of immune cells in slowly progressing and rapidly progressing models of cystic disease, and trying to understand if the immune environment is similar in these two different models to try to find a way to modulate immune cells to regulate the rate of cyst growth. So just real quick, for our lab, because I came from Brad's lab when he was a cilia lab, we mainly used the IFT88 model to study cystic kidney disease. I will show some data later on in other PKD models as well. But in this mouse model, we have the IFT88 gene, which is floxed around exons 3 to 4 of this gene, and it's crossed to a mouse containing a tamoxifen-inducible chicken beta-actin-driven Cre recombinase. So what happens is once tamoxifen is added to these mice, the Cre recombinase migrates to the nucleus, where it excises our gene of interest, the IFT88 gene, resulting in loss of IFT88 protein within about a week and loss of a functional primary cilia. So just keep in mind what I mentioned on that opening slide is if you don't have a functional primary cilia, you develop cystic kidney disease. Also keep in mind that the rate at which you develop cystic kidney disease in mice models, typically when you use this mouse model, is it takes several months to manifest cystic kidney disease. So like I said, our lab was really interested in trying to understand what's the role of immune cells in rapidly and slowly progressing mouse models of disease, and in particular in this IFT88 model. So what we did is we took a Cre-positive mouse, so this expresses the Cre recombinase downstream of that chicken beta-actin promoter, gave these mice, which we call the cilia mutants mice, we gave them tamoxifen. We waited 3 weeks to ensure that the IFT88 gene was deleted, the primary cilia was dysfunctional. And then 3 weeks later, we either gave them a 30-minute unilateral ischemia reperfusion injury, followed by harvest and analysis of the kidney tissue 2 months post-injury, or we simply took and induced these mice and then allowed them to age for about 6 to 7 months. Now, importantly, when we analyzed these mice at these two different time points, we didn't just pick these randomly, but the severity of cystic disease in these mice at the time points that we analyzed them was approximately similar. So what we're trying to do is we're trying to get mouse kidneys that had similar cystic disease, but had progressed at different rates to get there. Now, whenever you do single-cell sequencing, you always need to include a variety of different controls. So what we did is we took our cilia mutant mouse, we gave it a sham surgery. That just means we took the kidney out, but we didn't actually clamp off the renal artery in the vein. And then we allowed it to age for 2 months. So I will say that these mice were phenotypically completely normal, so they did not have cysts. We also took a Cre-negative mice, gave it tamoxifen, and then injured it to account for the effects of how a normal mouse kidney would respond to injury to see what the effect was there. And as the picture here would imply, we did not have cysts 2 months post-injury. And then last, we took a Cre-negative mouse, we gave it tamoxifen, and then we allowed it to age for 7 months to account for the effects that aging might have on the transcriptional signature, as it's been published in the past that aging can influence transcriptional signatures. So at the time points that I have listed up here, what we did is we sorted immune cells, epithelial cells, and all other cells from the kidney, and then we performed single-cell RNA sequencing. So for the purpose of today, I'm going to be focusing just on the immune cell part of this. So what we generated is what we call the immune cell atlas of cystic kidney disease. What I'm showing you here are UMAP projections from all 5 groups that I just described to you. So just for the purpose of understanding, the slowly progressing model will always be shown here on the left in the red. The rapidly progressing model will be shown here in the middle, and then the non-cystic control mice are going to be in black, as I shown here. So what we did is we simply started by going and say, hey, let's identify these different broad types of immune cells. So for example, we looked for clusters that were expressing T cell markers, CD3E, and we annotated these as T cells. Next, we went in and we quantified cluster abundance, and as you can see here, you don't really see too many differences between groups, and this is because for those of you who are immunologists, you know that immune cells have a lot of flavors. So there can be a lot of subsets, and we really like CDs and defining different immune cell subsets with those different CD markers. So what we did is we got out our magnifying glass, and we really wanted to zoom in to different cell subsets to see how they could be different between the 2 different models of cystic disease compared to one another in the non-cystic controls. So we did this for T cells, and what I'm showing you here is a UMAP from all the T cells that I just showed you in that previous slide. And now I think when you look visually, you can appreciate that the clusters of cells were really quite unique when comparing the slowly progressing model to the rapidly progressing model, as well as the non-cystic controls shown here. So next, we went to the literature, identified lineage-defining markers of these cell types, and were able to annotate all 13 different clusters of cells in this UMAP into the cell types that are shown here on the right. Next, we went in and quantified cluster abundance, and what you can appreciate is if you look at the data in the bottom right here, that we had 3 clusters of cells that were highly enriched in the slowly progressing model when compared to the rapidly progressing model and the non-cystic controls, and those were CD8 T cells, which is cluster 0, the T regulatory cells, which are cluster 1, and the Th17 cells, which are cluster 4. Likewise, when we looked at the rapidly progressing model, we had a significant increase in the number of naive and central memory T cells, as well as an increase in the number of effective CD4 T cells when compared to the slowly progressing model, although maybe not all the control groups here. But suffice it to say that we found that we had unique T cell clusters between each of these models. Next, and one thing that I always like to highlight when I give a talk is that you need to validate your data. So what we did is we went back in, we generated additional cohorts of mice, and came up with a gating strategy to identify all those different cell types that we just told you were different in our previous single-cell data set. So this is the gating strategy that we have up here on top for identifying all these different subsets. And to cut a long story short, we found that we're able to validate 4 of the 5 different clusters of cells. So we found that CD8 T cells and Th17 cells were really enriched in that slowly progressing model, and effector CD4 T cells and naive and central memory T cells were enriched in the injury-accelerated model of disease. So I've talked a lot about how these 2 clusters, how these different models of cystic disease were different. I think one of the next obvious questions you might have is, well, are they functioning to regulate the rate of cyst progression? So do they have a functional effect in disease progression? So what we did is we went back to our Cre-IFT88 mouse. We crossed it to a RAG1 deficient mouse, which for those of you who aren't familiar, RAG1 deficient mice lack all adaptive immune cells. So it may not be specific for T cells, but it does knock out all T cells in this model as well as B cells. So we crossed these mice to the IFT88 line and then induced them with tamoxifen. And then 3 weeks later, we either gave them that injury once again, followed by harvest and analysis 2 months post-injury, or we simply allowed them to age for 6 to 7 months and then analyzed the phenotype. And when we did this, we got a pretty surprising result. We thought for sure we'd have an impact in both of our models, but quite surprisingly, when we looked, we found that loss of adaptive immune cells and T cells substantially rescued cystic disease, but only in the injury-accelerated model. And you can see here quite clearly that in the slowly progressing model that didn't receive an injury, loss of these adaptive immune cells had absolutely no effect on the cystic phenotype. So it was something about the injury or the rate of progression possibly that was causing those T cells to be unique, that was causing them to be functionally important disease. So to summarize what I've showed you so far, in the slowly progressing model, we get an accumulation of CD8 T cells, we get an accumulation of CD4 Th17 cells. If you delete those cells, and we still don't completely understand why this is the case, but if you delete those cells, you have absolutely no impact on cystic disease. And we've tried lots and lots and lots and lots of different models. And suffice it to say that from what we've seen so far is loss of T cells in that model does not impact cyst progression. Likewise, in our rapidly progressing injury-accelerated model, we get an accumulation of naive and central memory T cells, as well as effector CD4 T cells. And in this case, if we lose those T cells, we substantially rescue cystic disease. So the next question you might have is, well, why exactly is that? What are those T cells that are enriched in that injury-accelerated model doing to regulate the rate of cyst progression? So in order to address this question, we went back to our informatics toolbox and we used this package called NicheNet, which for us, this is our favorite ligand receptor interacting tool. So what this tool does, and this is kind of the framework for how the tool works, it asks for what are ligands produced, are the receptors present, and then what are the transcriptional regulators and target genes downstream of those receptors that are activated in your cells. So to boil this down, in our injury-accelerated model, we thought that the T cells may be talking to the macrophage, which our lab has shown extensively, and I'll talk a little bit more in the future, that macrophages are really important for regulating cystic disease. So we thought maybe T cells are talking to macrophage to regulate cyst progression, or maybe T cells are directly talking to the cystic epithelium to somehow regulate that progression in the injury-accelerated context. So what this software does is it asks, what are the differentially expressed genes in the receiver cell? And in this case, it could be, once again, the cystic epithelium or the macrophage. And then it asks what ligands are present that could explain the DEGs in the receiver cell. Next, it asks, does that receiver cell express the receptor for the ligand? And finally, it prioritizes the ligands based on how many of the differentially expressed genes in the receiver cell that ligand may explain. So boil it all down to this, which is the plots that we get out when we put this in. What this data is showing is that this right here is the list of prioritized ligands that are produced by T cells crosstalking with macrophages in the rapidly progressing model. And the plot on the right is the ligands produced by T cells crosstalking with macrophages in the slowly progressing model. So what we did is we did a head-to-head comparison of this ligand receptor list and came up with a list of, I think there's 9 ligands that were specifically enriched in the T cells isolated from that rapidly progressing model. Furthermore, I'm highlighting interferon gamma here, and the reason I am is because interferon gamma is a known activator of macrophage. It induces a pro-inflammatory macrophage activation status, and it's also been shown to be specifically expressed by T cells. We have that data. I'm not including it here, but so what we did is we went back in and we looked for expression of interferon gamma in our different T cell subsets, and we validated that interferon gamma was enriched in this effector CD4 T cells, which if you remember, that was one of the 2 subsets that was highly enriched in the injury-accelerated model. Furthermore, we went in and generated another cohort of mice and analyzed flow cytometry interferon gamma protein expression. And similar to what we see at the transcript level, you get a lot of interferon gamma produced by these effector CD4 T cells. So the next question is, okay, you're telling me that interferon gamma is important for this injury-accelerated phenotype. So what we did is we went back to our pre-IFT88 line. We crossed these to interferon gamma deficient mice, and then we analyzed the phenotype at the same time points that I've been talking to later. So that's 2 months post-injury. And when we did this, we got a really striking phenotype, whereby what I'm showing you on the top, these are the interferon gamma control mice. These are the Cre-positive IFTD mice that get injured. And you can see here that this injury greatly accelerates the rate of cystic disease compared to the vehicle-treated mice here on the left. And that if we lose interferon gamma, we have a substantial rescue in the severity of cystic kidney disease. And this is shown here in the quantification of kidney weight, body weight on the left, as well as cystic index, cyst size, and the number of cysts. So this data, I think to us is pretty strong, indicating that after injury, you get that interferon signature, and that drives possibly macrophage activation and cyst progression. We also analyzed fibrosis in this model, and we found, and kidney function, and we found that once again, the injured mice, which are shown right here, they develop fibrosis. I probably need to get a more blown up image for this next time, but so my apologies there. But suffice it to say that they do develop fibrosis. And then when you lose interferon gamma, not only do you get fewer cysts, but you also get less fibrosis, which is shown in the quantification here. Furthermore, we analyze kidney function in these mice with BUN, and we have some creatinine data that's coming in as well. And we find that, once again, loss of interferon gamma substantially improved kidney function and reduced BUN levels. So all this data that I just showed you, this is in the IFT88 model. Once again, when we usually submit grants and papers, we typically get criticized because it's not an orthologous mutation, although there are IFTs that are mutated in ADPKD patients. So one of the questions we always get is, well, is this relevant to human disease and ADPKD patients? So we really are starting now to set out to thoroughly address this question. So what we did is, in collaboration with Dr. Darren Wallace at KUMC, is we're lucky enough to obtain some serum samples from either healthy controls or rapidly progressing ADPKD patients. And this is early stage ADPKD patients. This is prior to their end stage disease. So this isn't when their kidneys are failing. This is during the process of their progression. And what we did is we analyzed the level of interferon gamma in their serum. And when we did this, we found that those rapidly progressing ADPKD patients had significantly more interferon gamma in the serum. So we think that this suggests that there is some relevance to human patients here. And ongoing studies, I think we just got the samples in today, where we're going to be comparing the level of interferon gamma in the serum and urine of slowly progressing ADPKD patients compared to rapidly progressing, to really, I think the goal is at least to really hammer home the point that the rate of progression is really driving this subset. So just to summarize the first part of my talk, these interferon gamma positive T cells, we think that they promote injury accelerated disease. And we also showed that ADPKD patients that are rapidly progressing have more interferon gamma in the serum compared to healthy controls. This is just highlighting some of the future studies that we're looking at, which is trying to test if this is a macrophage dependent phenotype. So trying to see if these are signaling through the interferon gamma receptors present on the macrophage. So we're making some cell type specific knockouts of this receptor in macrophages to really get after that point. So next, I want to take you back to our single cell atlas here to zoom in on another cell type, which is honestly is probably about 75 to 80% of what we do in our lab, which is macrophage biology. So this time, instead of zooming in on the T cells, we will zoom in on the myeloid cells. And when we subsetted our macrophages, or mononuclear phagocytes, I should say, once again, we went in, we compared the different groups shown here on the top in the UMAPs. We annotated genes based on canonical marker expression. And then we quantified cluster abundance in our biological replicates down here in the heat plot table on the bottom. And when we did this, we found, kind of similar to what we found with the T cells, that each model of cystic disease, even though they had the same genetic mutation, had similar cystic index at the time of analysis, each model had unique macrophage subsets. So for example, the rapidly progressing model had enrichment of what we call the dedifferentiated kidney resident macrophage. And we called it this because it had canonical kidney macrophage markers, but it also lacked some of them as well that are typically found on these macrophages. And we also found this population of MRC1 positive kidney resident macrophages that were enriched in the slowly progressing model. So once again, the first thing we wanted to do is we wanted to validate our single cell data. So we went in, we designed a panel to identify the dedifferentiated kidney resident macrophages, and this is the gating strategy that I'm showing you here. Suffice it to say, when we identified CD64 F480 double positive kidney resident macrophages, we found that, unfortunately, and it's not the best way to probably do this, but we found that the dedifferentiated macrophages could be identified based on the absence of CD11C expression in the kidney, even though they still expressed F480 and CD64. So what we did is we went back into our rapidly progressing model of injury-accelerated disease, because this is the one where we found this population enriched, and then injured these mice, harvested them at different time points after injury, and then quantified the number of dedifferentiated kidney macrophages. And when we did this, I think you can kind of appreciate in this backplot shown in here in the upper left. But what we found is that beginning 7 days post-injury, we actually got a strong accumulation of these dedifferentiated kidney-resident macrophages shown here. And just to highlight that this is the time of first cyst initiation in this model. So we think that, and all the data that we've collected over the years suggests that these macrophages are really important for regulating progression. From what we found, they're not involved in cyst initiation, but they're simply regulating how fast the disease progresses. So in quite a bit of work that I'm not going to go into today, we found that in these cilia mutant, these IFT88 mice that got injured, what happened was that following injury, they significantly increased their expression of the membrane-bound form of CSF1, which, for those of you who aren't familiar, that basically means that these epithelial cells kept the CSF1 present on the surface of the epithelial cell rather than secreting it into the environment. And this led to paracrine activation of these kidney-resident macrophages, which are directly adjacent to the tubular epithelial cells, leading to their self-proliferation and accumulation of that dedifferentiated phenotype. So we think that proliferation is really one of the factors that was driving that accumulation of that population. We also showed in some parabiosis and some fate mapping studies that I'm not going to go into today that they were not a monocyte-derived population, but rather they're accumulating through self-proliferation. So in order to test this hypothesis and really prove our point, what we did is we went to the literature and we found the CSF1R kinase inhibitor, which specifically prevents downstream signaling from the CSF1R signaling axis. And we gave this to our rapidly progressing mouse model to see what effect that had on macrophage number. So what I'm showing you here is data looking at the number of total kidney-resident macrophages, as well as the number of dedifferentiated kidney-resident macrophages. And what we found was very nicely, when we gave the CSF1R kinase inhibitor to prevent that proliferation, we substantially reduced the accumulation of those dedifferentiated kidney-resident macrophages. And keep in mind, this was specifically in our rapidly progressing IFT88 model. Furthermore, when we looked at the phenotype, we found that preventing those dedifferentiated kidney-resident macrophages from accumulating was able to substantially rescue the severity of cystic disease. And we also showed this in a second model as well. So it wasn't just specific to the IFT88 model, but it seemed to be true for multiple models of PKD. So to summarize the second part, we find that these dedifferentiated macrophages, which we identified via single-cell RNA sequencing, were accumulating in a rapidly progressing model of injury-accelerated disease, and that these dedifferentiated macrophages produce inflammatory mediators. I didn't show any of this data today, but we showed that they produce IL-6, TNF-alpha, IL-1 beta, and we think that that causes damage to the tissue and that causes the disease to accelerate. And then if we prevent them from accumulating using that kinase inhibitor, that we rescue the disease. Sorry, I'm getting over a cold. Next, taking you back to our UMAP atlas here. We talked a lot about this dedifferentiated cluster right here, which we showed promoted the disease progression in our rapidly progressing model. Next, I want to take you back and address the role of these MRC1 positive cells in the slowly progressing model. So one of the things we found was that when we did a differential expression analysis looking for genes that were highly enriched in these MRC1 macrophages, one of the things we found was that these cells, one of the genes that was most highly enriched in this cluster was TREM2. And I don't know if anybody in here is a macrophage biologist, but if you are, this should set off alarm bells in your head, because of some of the seminal work done by Ido Amit and Marco Colono and others, showing that this TREM2 macrophage signature was very unique gene signature that was accumulating in different disease models. So what I'm showing you here is the Karen Schall paper from Ido Amit's group, and it was published in 2017, showing that in an Alzheimer's disease model, you got accumulation of what they call a TREM2 positive DAM, or a disease-associated microglia. So you can see here, this is a UMAP looking at all of the microglia cells, and you can see here that in wild-type mice, whether they had TREM2 or did not, all these microglia could be found in the homeostatic microglia cluster shown right here. Now, in contrast, when you induced Alzheimer's disease in a TREM2 proficient mouse, you can see that these microglia slowly transdifferentiated into a TREM2 stage 2 dam, or disease-associated microglia. In the contrast, if you lost TREM2 expression, it prevented the accumulation of that stage 2 dam and prevented the accumulation of that dam signature. So this was like probably one of the papers that inspired me to get into the immunology field, was some of this work by Ito Amit's group showing that you needed TREM2 for accumulation of these TREM2 macrophages. And it really turned out to be kind of fortuitous, because TREM2 is one of those genes that was really highly enriched in our model. So in order to study the function of TREM2 macrophages in cystic disease, we're fortunate enough to have a collaborator at OUHSC that was working with Marco Colonno at the time. So what we did is we took our Cre-IFT8 mice. So once again, these are the same mice that I have been talking about the whole time. And this time what we did is we crossed them to a TREM2 deficient background. And then we induced these mice with tamoxifen, and then we waited approximately 7 months to harvest and analyze this tissue. So once again, this is the IFTA model, but this is the slowly progressive model this time. So when we did this, one of the things that was kind of interesting to me is that I kept getting these notices from the animal facility that your mice are dying. It's like, what is going on here? I don't understand why they're dying. And it turns out that when we lost TREM2 in this slowly progressing model, our mice seem to be dying at a much faster rate. So approximately 1/3 of all of our TREM2 deficient mice on the IFT8 background were dying prior to the harvest point. Whereas you can see here, the mice that are either TREM2 heterozygotes or TREM2 wild type and cystic did not have that phenotype. Now, if we take a look at the mice that did make it to our harvest point, and you look at histologically what they'll look like, you can see that the IFT8 mice that lacked TREM2 had much worse cysts compared to the mice that had TREM2. And that's shown in the quantification here on the right. So based on these data, we hypothesized that those TREM2 macrophages may actually be restricting cystic disease, which was the same phenotype that was published by Charlie and Ito a few years ago. So to further test the importance of these TREM2 macrophages in disease progression, we actually took advantage of kind of a fortuitous finding that we had in our lab that we published last year, whereby we found that when we knocked out CX3CR1 in analyzed kidney macrophages via single-cell RNA sequencing, we found that the absence of CX3CR1 actually caused the induction of a TREM2 macrophage signature. So you can see here, the TREM2 cluster is the one shown here in orange. You can see in our quantification of biological replicates on the right that this cluster is really highly enriched in our mice lacking CX3CR1. And you can see from the UMAP and the feature plots shown here on the bottom that the TREM2 expression is really highly enriched in this cluster. Now, if any of you guys ask me why this is, I'm going to tell you I have no idea. I don't necessarily know why this is happening, but it just turns out that the biggest effect that we saw in the absence of CX3CR1 was the accumulation of a TREM2-like macrophage signature. So luckily, because we see an accumulation of a TREM2 signature in these mice, we can apply this to our IFT8 model to see if you accumulate TREM2 macrophages, how does that impact cystic disease? So what we did is we took our CX3CR1 deficient mouse, crossed it to the IFT8 line, and then we gave them the injury again to recapitulate that rapidly progressing model that I was talking about earlier. And when we did this, we found that induction of a TREM2 macrophage signature in the IFT8 injury accelerated mouse, so once again, these should be rapidly progressing mice, almost completely prevented cyst progression or cyst formation. So, and I will say this is one of the more striking phenotypes that I've ever seen in the PKD, in my time in the PKD field, where I analyzed all these mice myself, and I barely saw maybe 1 or 2 cysts per kidney in these mice. So it was a really striking phenotype, and I think it, at least at some level, supports our idea that those TREM2 macrophages restrict cystic disease. Now, we also crossed the same CX3CR1 deficient mouse to another mouse model of PKD. So in the PKD field, we're also great for having a lot of different models to mimic the human disease. And that's because I think what I mentioned in my earlier slide, there's a lot of genes that can be mutated that can cause the human disease. So what we did, like I said, is we crossed this to a CAG-CRE IFT8 PKD2 model, which is directly orthologous to AD-PKD patients. And when we did this, we saw the same effect that we got in the IFT8 model, whereby induction of a TREM2 macrophage signature was able to substantially reduce the severity of cystic disease in these mice. So in summary, the TREM2 macrophages appears to really restrict cyst growth, at least based on the preliminary data that we have so far. So I'd like to finish up in my last few slides by just talking about how whenever we submit papers or grants, we always get this criticism that all this data that you just collected, and the majority of what you just showed me is in the IFT88 model, is this data that you showed and these macrophage signatures, are they relevant to other models of PKD? So do you find TREM2 macrophages in models that are directly orthologous to those AD PKD patients? So in order to address this question, we've spent the last, I'd say probably 6 months to a year generating the single cell atlas of PKD, whereby we incorporate all of our unpublished data and all of the published data from our lab and other labs into a single comprehensive atlas of PKD. And I'm just showing you up here, this is the data just looking at kidney macrophages in all the different models of PKD. So I'll walk you through this. So what we have here, this is a clusters of kidney macrophages. You can see we have 4 different clusters, including homeostatic kidney macrophages, TREM2 kidney macrophages that I was just talking to you about in the past, as well as those dedifferentiated kidney macrophages that I've also been talking about. This right here is a UMAP based on whether the cell was from a control or a PKD sample here. And then on the right, this is really kind of the take home of all the different models that we incorporated into our single cell atlas. And you can see, it's a little bit easier to see up there than it is on my screen, but the top 2 models, this is our slowly progressing IFT88 model. The one below that, that's our rapidly progressing injury-accelerated IFT88 model. This one right here, you can kind of see it better this way. The PKHD1-CRE, PKD1-FLOX-FLOX, that's another really rapidly progressing orthologous model. The PKD1-RC, RC is a very slowly progressing orthologous model. And then the CRE-positive PKD2 is also a slowly progressing orthologous model. So what I did is I color-coded these groups based on whether they're rapidly or slowly progressing. So all the ones that are shown in blue are slowly progressing models of PKD. All the ones shown in red are rapidly progressing models of PKD. Now, I know it's a lot to look at. There's a lot of groups. Don't worry about that. The thing you need to really focus on is these quantifications shown here in the bottom. So what we did is we quantified the number of cells in control versus PKD groups. So if there is more cells in the PKD group, it would be higher and shown further to the right. If there's more in control, it'd be higher to the left. And what we found, long story short, is that there was more TREM2 macrophages and more dedifferentiated kidney resident macrophages in multiple PKD models compared, and multiple slowly progressing PKD models compared to the rapidly progressing models and the non-cystic controls. This right here, this is looking at the number of cells based on whether it's a rapid model or a slow model. And you can see here, once again, we found more TREM2 macrophages in our slowly progressing model, and we found more dedifferentiated ones in the rapidly progressing model. Now, if we take and we zoom in, we just wanted to make sure that that difference wasn't driven by a single model. And what I'm showing you here is the number of TREM2 macrophages based on the individual models. And I think you can appreciate that all 3 of our slowly progressing models had more of those TREM2 macrophages than did either of the rapidly progressing models shown here. And this on the right, this is another heat plot, just or a frequency plot, just looking at the proportion of TREM2 macrophages in each individual model. So slow models are shown here on the left. Rapid models are shown directly adjacent, and then all of our non-cystic controls are shown here on the right. And you can see, I hope you can appreciate that the TREM2 macrophages are definitely enriched in the slowly progressing models. So next, what we did is we wanted to validate this data once again. So we went in and this took quite a bit of effort. We generated 4 additional models of rapidly progressing PKD, and did 2 models of slowly progressing PKD, and then we quantified the number of those TREM2 macrophages via flow cytometry. So this is at the protein level. And when we did this, similar to what we found with our single cell atlas, that the number of TREM2 macrophages was significantly enriched in our slowly progressing model compared to our rapidly progressing models and our non-cystic control. And this encompassed not only the rapidly progressing models I showed you in the previous slide, but additional rapidly progressing PKD2 models. So don't necessarily worry too much about what the model was. Just suffice it to say that slow models, more TREM2 macrophages compared to rapid models and non-cystic controls. So the last thing I'm really going to talk about today is people will say, okay, well, you found all these clusters in these population of mice. Well, is this relevant to AD-PKD patients? And what's the translational value of your findings? So we set out to address that question, and we did that by generating a combined single cell atlas from control and AD-PKD kidney tissue. So what I'm showing you here, this is once again a UMAP looking at different looking at different kidney resident macrophage clusters. So these are all different flavors and subsets or states of kidney resident macrophages. And similar to what I showed you in the mouse single cell atlas, we get homeostatic kidney resident macrophages. We get our TREM2 macrophage cluster, and then we get our dedifferentiated kidney resident macrophage cluster here. Now, one of the issues that we have, and if anybody has data or ideas of where to get data on, we have a limited number of control cells here. So the KPMP doesn't have a ton of those cells that I can find at least. But if anybody has a lot of macrophage single cell data in human kidneys, I'd love to talk to you or collaborate more with you. But suffice it to say, once again, when we quantified the number of these macrophages in control versus PKD tissue, we found that the TREM2 macrophage and the dedifferentiated macrophages were really substantially enriched in the AD PKD tissue compared to the controls. Something that we found really interesting, and I always like to bring up, is that AD PKD is a really heterogeneous disease. So despite the fact that they have cysts, it's been shown recently by some imaging studies that some of those cysts progress really rapidly in human kidney tissue. Some are really stable, and some actually regress. So the nice thing about the data that we have here is this is from 7 individual cysts from a single AD PKD patient. So what we can do is we can assess the intra-kidney heterogeneity of these AD PKD patients. And when we did this and we quantified the number of TREM2 macrophages versus dedifferentiated macrophages in these in this AD PKD patients, what we found was that some cysts had a lot of TREM2 macrophages, some cysts had a lot of dedifferentiated macrophages. And I think our working model would be that the cysts that have a lot of TREM2 macrophages may be progressing more slowly, whereas the ones that have more of those dedifferentiated macrophages, which once again, they're enriched in a rapidly progressing model, we think maybe around those rapidly progressing cysts. We also went to Dr. Ben Humphreys, generated a single nuclear RNA sequencing atlas of AD PKD, and we showed that in his single cell atlas that we also found an enrichment of these TREM2 macrophage signature genes, so TREM2 and MRC1, which is highly enriched in his leukocytes from AD PKD patients shown here compared to the controls, whereas those dedifferentiated macrophage genes were enriched in some PKD samples, but not all. So you can see that the PKD didn't have any expression of this gene, whereas this did. And then one of the last things we did is we went in and we once again went to human tissue, and we wanted to validate our single cell RNA sequencing findings. So we did some flow cytometry in human AD PKD kidney tissue, and we're able to validate that we're getting more TREM2 macrophages in AD PKD tissue compared to control. So we think that what we're finding in the mouse is pretty relevant for AD samples. So to summarize everything that I've just talked about, what we find is that in the rapidly progressing models of PKD, you get an accumulation of interferon-producing CD4 T cells. We think that they promote, possibly, although we don't necessarily know how or if interferon gamma is directly involved in this process. We haven't really analyzed that in a thorough way. We think that it is associated with the accumulation of these dedifferentiated macrophages. We've published in the past that these dedifferentiated macrophages produce IL-6 TNF-alpha, which drives cyst progression and rapidly progressing disease. In contrast, in the slowly progressing model, we get an accumulation of CD8 T cells, maybe T regulatory cells, although the flow cytometry data may call that into question. Th17 cells, we think that this microenvironment, these T cells may be producing something that affects the accumulation of those TREM2 macrophages, although we get rid of those T cells that didn't have a phenotypic effect. So we're kind of not sure, to be honest. We don't know. But anyway, you get an accumulation of TREM2 macrophages, and then we think that they can act to restrict cyst growth by creating more of an anti-inflammatory environment and phagocytosing those apoptotic cells. And then last, we show that if we induce a TREM2 macrophage signature in the rapidly progressing model using that CX3CR1 deficient mice, that we're able to prevent cystic disease through accumulation of those cell types. So last, I'd like to finish by thanking the people in my lab. I'd like to thank my collaborators. In particular, I'd like to thank Dr. Katarina Hopp at Denver for all of her help with the T-cell project. I'd like to thank Florent and Mary Beth for their collaborations in the macrophage projects, Dr. Chin Ma and Anjun Ma at Ohio State for the help with some of the computational work that I'm showing you here, and then my current funding sources. I'll be willing to take questions. Thank you.
If you have questions, talk into the mic.
Thanks, really nice presentation. So the question I have is, what drugs does this suggest? Do you have ideas for drugs? Like, what mechanism would you go after? And I guess that to back up, maybe I missed this, but can you tell ahead of time which patients would be slow progressing versus rapid progressing? Because it makes all the difference, it sounds like, in the choice of drugs.
Yep.
So are there candidates that you're considering? And And I guess there could be a lot of side effects too, because you're affecting the immune system.
That's yeah, that's like 5 really great questions. So are there things that predict the rate of cyst growth in patients? The answer to that is not really. I think our kind of like hope and idea would be that what we're finding here could be a way to find those things that would predict the rate of growth. So for example, we found that in if we looked at serum from AD-PKD patients, that the ones that were rapidly progressing had more interferon gamma than the healthy controls. Now, the ideal experiment would be comparing the rapidly progressing to the slowly progressing ones and show that the rapidly progressing ones have more interferon gamma, and that predicts future rate of decline. So the nice thing about the study that we have there is that is AD-PKD patients that have normal kidney function. And what they do is they're actually tracking them over the course of several years to be able to kind of address that question. So we're analyzing both interferon gamma and TREM2, a soluble TREM2 expression in those models to kind of try to get predictive biomarkers. But yeah, that's like a gold, that's like the golden goose question is if we can identify those markers that predict the rate of progression, that'd be a really big help for the field. As far as drugs, I think you hit the nail on the head. Finding a drug that targets the right person at the right time would be really important. So I guess our model would be that is if you have a patient that is rapidly progressing, we'd predict that giving them like an interferon gamma blocker, such as like gammaphage or something like that, would prevent their disease from progressing more rapidly. Now, if you have a slowly progressing patient, it's not as big of a need, but all these patients, for the most part, will end in end-stage disease. So if we can take a slowly progressing patient and make them progress even slower, that could be really therapeutically beneficial. So for those patients, we would think that maybe targeting TREM2 activating antibodies would be kind of like the way to go for that intervention. But yeah, I think finding biomarkers that predict the rate of progression can be really important. I think that's something that would be a really great finding for the field and would really help guide our immune studies, because you're right, targeting the immune system can be really tricky. So for example, like we just published a paper last year, I think it was, where we did checkpoint inhibitors for PKD patients. And we found that if you give like a combo of checkpoint inhibitors for AD-PKD patients, it will restrict disease progression. But people, as people know, like giving checkpoint inhibitors for a really slow progressing disease is just kind of like a crazy thing to try, right? Because you can give checkpoint inhibitors for cancer patients because they typically don't have that long to live. But AD-PKD patients, even without any therapeutic interventions, can live for decades. So I think trying to find very specific targets of disease modalities will be important. And the nice thing is those TREM2 macrophages, from what we can tell, are really, really specific for disease. Like we pretty much only find them present when there's some sort of kidney disease present, whether that be PKD, we have data in IRI, we have data in DKD, that same signature is only present when the disease is there. So I know that's kind of a long answer, but yeah.
So basically, you're telling us this the TREM2 macrophages and should be very stable and stay in a disease kidney, right? So you're not going to see them moving around.
No, to my knowledge, they should not be moving around. No.
Okay. So if you look at by imaging and what kind of distribution you can find?
Of the TREM2s.
Uh-huh.
So if we look in mouse or human, we've done that with RNA scope. We find them around the cysts. So they're so if you look in a human section, the cyst will be like here, and the TREM2 macrophages will be around that cyst pretty nicely. So it's very cyst localized.
Thank you.
Great talk, Kurt. So tons of questions, but let's just focus on a couple of things. When you did the experiment with the RAG knockouts, right? So I was also struck that Tregs were enriched in the slow progressors. So what do you think about that? You were kind of focused more on the effector T cell populations. What are the antigens that these immune cells are actually recognizing as well?
Yeah, we have no idea.
Okay.
I don't know. We've thought about that. We've been asked that a lot in the past. We thought about doing like TCR sequencing to try to get at that. There's some data at least in the injury accelerated model, that might suggest it's a polyclonal T cell response, whereby they're not responding to a specific antigen, they're kind of responding to like kidney damage. So Hamid Rab's group showed that there's a polyclonal T cell response to kidney injury. We don't know in our model, but we think it's a polyclonal, not a specific antigen.
And then I think following up on, I think, Erica's question about interventions, right? So the interferon gamma knockout, the histology that you presented was kind of striking because the cortical cysts were reduced, but there were kind of juxtamedullary regions that were still quite cystic. So that's kind of interesting in its localization based on your own work as well on the regional heterogeneity of macrophages, right?
Sure.
And then the other point related to the intervention is the CXCR3, is that what, fractal kind? I don't know what
Yeah, yeah.
So wouldn't you predict that, are those levels higher in patients or in the mice? Would those, I mean, can you actually modulate those levels to affect the levels of the TREM2 macrophages?
Yeah. So the first question is regarding the interferon phenotype. Yeah, it's a really peculiar phenotype. So the one thing that we've really noticed is that in the mice, the Cre positive that we injure, the knockout for interferon gamma, I'd say about 2/3 of all the cysts that you find, if you really zoom in, are glomerular cysts. And it's you also get some in the juxtaglomerular kind of like region, but if you really zoom in, you get a lot of like glomerular cysts where the glomeruli seem to like sunk a little bit further down from the cortex. I've noticed that as well, and I honestly I have no idea. It was a weird thing. There's some literature out there that's pretty old that shows if you knock out interferon gamma, you can develop cysts in the kidney. Our pure interferon gammas by themselves, we don't really see that in our hands, but maybe there's something about interferon gamma being needed for preventing that. I honestly I can't really tell you why. Regarding your second question, so we've looked at CX3CR1 in the mouse, and we do see that expression is up in PKD. I'll be honest, I haven't really thought about modulating CX3CR1 expression to regulate or to induce a TREM2 signature. I'm not, I think the reason we haven't really thought about that was because we're not exactly sure why they come about. That whole kind of like outcome that we got was really kind of surprising. And I was talking with Dr. Crisp a few, like half an hour ago, and I was telling him about this phenotype, and I was saying, we have no idea why you get that phenotype. It's something, if we understood better how those TREM2 macrophages were accumulating in that CX3CR1 deficient model, we might be able to think about addressing it that way. But I think for us, it's kind of like a dream. So I think we'll kind of have to wait to figure out why. We just don't know. So. Go ahead.
So I'm going to go all the way back to the beginning
Okay.
because I don't understand something.
Okay.
So if I remember correctly, you said that this was, people inherit one dysfunctional copy and then they get a second hit.
Mm-hmm.
So it's a predisposition, basically.
Yeah.
Right.
Mutation, yeah.
So that would mean that you get the second hit, which I assume is a second mutation. That is it a second mutation?
Presumable.
So if that's the case, then the amount of genetic heterogeneity in your kidney must be massive, right? Because you're getting cysts all over the place.
But yeah, it's thought to be clonal within the cyst.
Within the cyst.
Outside of the cyst in PKD patients, we don't really know, but it would be anticipated based on what I've been yammering on about, that yes, that you get a lot of heterogeneity there.
So there's this whole thing now in somatic mutations, accumulation in basically like cancer associated genes, the things that cause proliferation. Doesn't lead to cancer necessarily, but
right.
And so do you think that's kind of like what's going, you're getting this mutation and you're getting this proliferation. It's worse in this case because you're hitting the gene that.
Yeah, I mean, I think that there's
Oh, I guess let me,
We don't know.
Yeah.
Nobody's really like,
nobody's sequenced any of those.
nobody's done a sufficient enough job of sequencing that I feel confident in that answer.
Okay. And then, so the follow-up question to that is that your exposure is IR or
Ischemia
radiation,
reperfusion,
right?
Yeah, ischemia
Oh,
reperfusion.
ischemic reperfusion.
Yeah.
Okay. Never. So that's non-mutagenic, which is that's or presumably non-mutagenic.
Mhm.
Have you done any sort of like
In the mice? Or-
yeah,
well,
like mutational exposure, like
Uh,
you gave a mutagen.
Yeah, so in the mice, it's a genetic knockout.
Yeah.
So it's a little bit different than humans. So if you take a mouse that is a heterozygous for PKD, like a human patient, and you age them to a year, they don't have cysts. Don't know why. If you age them out to 2 years, they develop a few small focal kind of like cyst-looking things. We don't necessarily completely understand why the mouse and the human are so different in that context. I think like what our lab would probably argue is that it's some environmental exposures that maybe cause like somatic mutations in random, it's even I don't know, it's even possible that it's not even the same gene. It could be another ciliopathy related gene. So there's some data from a guy at UT Southwestern that I'm friends with that he's kind of blowing my mind about how all this works, because we always kind of assume that you have one dysfunctional copy, you have a mutation in that second copy, and then you basically lose expression of that gene, right? That's kind of like the paradigm I grew up on in PKD. Well, he's actually showing that if you give a mirror, a microRNA, that you can re-express PKD in PKD1 deficient cells. So, that's clinical trials. It's in like phase 3 clinical trials right now. So they've shown that it increases PC1 expression in the urine, and that there's positive trends in the height adjusted total kidney volume data that they have clinically from AD-PKD patients. So that kind of that data, and we can talk more about it at dinner, but that kind of like blew my mind, and I'm still like, I have a hard time wrapping my head around how that really works, because like I said, we typically think that you have one dysfunctional copy where you're not. It's like PKD is thought to be a haploinsufficient, and it's thought to be a dosage disease, right? It's like you need this much functional polycystins to not get the disease. If you dip just a little bit below it, you start to get the disease. So I don't know, that whole concept. I don't really have a good answer for you. I don't know. My head is just, it kind of spins when I think about that, to be honest. We can chat more later.
Just out of curiosity, have you tried tolvaptan? Does anybody know how that works? I've heard that nobody really knows how tolvaptan works,
no,
and it doesn't work very well. And I don't know if there's any mechanism here that you've looked at.
Yeah, tolvaptan definitely works. It's a vasopressin receptor antagonist. It works on distal tubule cysts, prevents cell proliferation, fluid secretion. That mechanism's, I don't feel confident saying a lot of things, but I feel pretty confident saying that mechanism has been fairly well sorted out. We've thought about it. We haven't done anything with vasopressin in our hands, looking at immune cells or anything like that. Yeah, the mechanism is known though. Vasopressin does seem to work clinically. It slows the rate of progression, and it prevents fluid secretion, it prevents proliferation. It seems to hit both of those kind of hallmarks of PKD. So,
But patients may need more than that, like the mechanism that you're
so,
looking at.
So tolvaptan works, but if you look at like survival rates and basically like kidney disease parameters, what happens is like a normal, well, I don't want to say normal, but a PKD patient, if you look at GFR, will go down like this. And if you look at one with tolvaptan, it'll go down like that. And the difference between the tolvaptan and the non-tolvaptan group is probably 3 to 5 years. So it definitely has a substantial effect on disease progression. It certainly prolongs their course, but as you kind of like, they both still go down. They both still will reach end stage disease. And I think one of the things clinically that they talk about a lot is trying to decide what cohort or what subset of patients is best for giving this drug to. So like kind of what you mentioned to me, there's rapidly progressing, there's slowly progressing. Do we give it to one? Do we not give it to other? And I talk to a lot of clinicians, and that's still something we don't really understand or know. So yeah, I have one online. Yeah, so the question online is, can can you do organoid models of PKD, which presumably lack a significant immune component, also demonstrate features of rapid or slow progression? So I think they're trying to ask, if you do organoid models, can you recapitulate the rapid and slow progression? I am not sure. So Bino Friedman, who works here, would be able to better answer that than me. But I'm presuming they probably can modulate that rate of growth, although it's probably not as extreme as what we find in the mice. I don't know the answer to that. That's a good question. What? Okay. Okay. Yeah, that's yeah. And I'm not sure if like that rate of growth is quite as drastic as what we find in the mice. So in the mice, we get like really pretty drastic rates of cyst growth and cyst progression. I'm not sure. I'm not sure. I'm not sure how well the organoids can model that. But yeah, I guess shear stress. Yeah. It's I talk with some of the organoid people quite a bit about trying to put the macrophages in that, and I think people are doing that right now to try to see how the macrophage can modulate cyst growth in the organoids. But yeah, I'm not sure completely. All right, well, thank you guys.